Hello, I'm Robert LeBoyer, Senior Biotechnology Analyst at Noble Capital. With us here today is Atara Bio with Eric Heiligren, the Executive Vice President and Chief Financial Officer, Jason Awe, the Head of Investor Relations, and Dr. Koki Nguyen, the Executive Vice President, Chief Science and Technical Officer. Dr. Nguyen will give a presentation, followed by some questions and answers. Dr. Nguyen, please go ahead.
Thank you, Robert. Thank you for your time. At Atara, we're the first company to deliver on the transformative potential of allogeneic T-cell therapy. What that means practically is we're the first company to obtain regulatory approval for an allogeneic T-cell immunotherapy. Ebvallo was approved in the EMA December 2022 in our partnership with PureFab. We also expect to file our BLA soon in the United States for approval in the United States. We have near-term milestones with ATA3219, which is our differentiated allogeneic CD19 CAR T-cell therapeutic. We're going to walk you through why we are excited about this program and talk through why this program contains clinically validated technologies. We filed an IND with the FDA, not only for large B-cell lymphoma, but also for lupus nephritis. We anticipate clinical data in the first half of 2025. And we are initiating our large B-cell study in non-Hodgkin's lymphomas, and we anticipate initial clinical data in the latter half of this year. We're at a momentous time in oncology as well as autoimmunity. Autologous CAR T therapeutics have shown amazing responses in patients. These patient-derived cells have shown consistent efficacy, and physicians know how to deal with the side effects of autologous CAR T cells. However, we would say that there's two problem statements that we're trying to solve at Atara. Number one, can we increase the efficacy that's seen with these autologous CAR T therapeutics? And number two, can we make the CAR Ts in a more economic way to get these therapeutics to patients sooner? And that's the promise of allogeneic cell therapy. You get T cells from healthy donors, you manufacture them ahead of time, and then you have these amazing therapeutics ready to deploy and ship to patients within days. And because of how these T cells are made, which is with healthy donor T cells, and because we can build up the inventory, we think that this can transform this already exciting therapeutic modality. Let me give you a little background for our platform. So our next-gen CAR T platform is built on the chassis of Ebvallo. And there's a lot of advantages for this, we think. The first thing that you should know is that Ebvallo it's a T-cell therapy that recognizes EBV-infected B cells. There's no gene editing of the T-cell receptor or MHC molecules, and we utilize the principles of transplantation. So we do partial HLA matching, so we match 2 of 10 alleles. There's no lymphodepletion, and we have robust manufacturing for Ebvallo. We've also established a global supply and logistics process. This is what is approved in the EMA. Now, you can imagine when we evaluate what we learned from Ebvallo, it follows that we feel like this is a great starting point for a next-gen allogeneic CAR T-cell therapy. We are going to retain all of the features of EBV T cells, all of the efficacy and safety we've seen in almost 500 patients, but we're going to stay true to the biology of T cells. So we're not going to introduce complex gene editing. We're also going to include next-gen chimeric antigen receptor designs. And we're going to talk to you in the next slide about the key features and then tell you why we think these key features have been clinically validated. Before we get into our platform in detail, we wanted to tell you about our pipeline. We feel like we have a robust, differentiated allogeneic T-cell immunotherapy pipeline. We've talked about tab-cel. Again, we expect the BLA to be submitted soon. It's already approved in the EU. And today we're going to talk to you in depth about ATA3219, our CD19 targeted CAR T-cell therapy for non-Hodgkin's lymphoma, as well as autoimmune diseases. We'll also touch upon our second CAR T program, which is ATA3431, which is our dual CAR program targeting CD19 and CD20. When you consider tab-cel, this BLA submission is on track for Q2 2024. We have a strong clinical file. We had a positive pre-BLA meeting with the agency, and we have historical data for why we're confident about this product in the United States. Well, for over 20 years, we have a consistent response rate of 50% in a patient population that is really, they have no recourse. These patients without tab-cel basically could die within weeks and months. Tab-cel has a favorable and safety profile. So even though it's allogeneic T-cell therapy, we haven't seen any graft-versus-host disease and any other adverse events related to tab-cel. We know now, over the development of this program, there's a consistent duration of response and overall survival rate. And again, this product has been used in almost 500 patients. Not only have we seen this tremendous efficacy over time, this has actually been recognized. The agency has granted this breakthrough therapy. It also has orphan drug designation. And again, the reason why we're taking this to the finish line is the patients are waiting and there's no other treatment options available. In addition to tab-cel, which is really at the commercial scale, we are now pivoting to our CAR T platform. Our CAR T platform uses the natural biology of T cells and all the lessons we learned from commercializing tab-cel in the EU. But let me tell you a little bit about the key features and how this ATA3219 program is differentiated. The first key element is that we retain the T-cell receptor. We feel like this is a key difference. There's a lot of allo CAR T programs which may edit out the T-cell receptor. However, we know this is a key survival signal for T-cell biology. The second is the EBV specificity of our product, we think also contributes to the safety of this allo CAR T therapy. So what we've learned from tab-cel is with our EBV targeted T-cell receptor repertoire, we have not seen any off-target toxicity. And we think that's because this EBV specificity is obviously very specific for viral antigens. And over time, we've looked, but we have not found any adverse events. In other words, we feel like this is safety built into our CAR T product. The next important feature is that we've retained the MHC in our product. And we think this is important. While a lot of other approaches may employ genetic editing of the MHC molecules on the T cells, we feel like this is key to our allo solution. Partial HLA matching worked for tab-cel, and we think that it's going to work for our CAR T program. This enables an allogeneic approach that avoids host versus graft rejection. The last two key aspects of our T cells is we have a less differentiated phenotype. So our alpha beta T cells are manufactured with purpose, and we engineered a central memory phenotype. And we're going to talk about that with the data. We think that this has also been clinically validated, as well as the way that we design the chimeric antigen receptor, which employs the 1XX costimulatory domain. Again, we feel like all of these elements make this a differentiated CAR T program that we're very excited to put into patients this year. I mentioned this on the last slide, but this is to bring home the point that clinical data from industry leaders as well as academia reinforce these key attributes that we've engineered into our CAR T program. The first is the importance of this EBV CAR T platform. Memorial Sloan Kettering did an initial investigator-sponsored study with EBV CAR T cells targeting CD19 positive malignancies. In this study, the overall response rate was 83%, and 5 out of the 6 patients had a durable response that lasted greater than 2 years. We feel like this is pretty powerful validation for EBV CAR T cells. But then when you add on the second and third elements, we're even more positive about our program. The 1XX co-stim domain has been used in the clinic. This has been employed by Takeda, collaboration with Memorial Sloan Kettering. Takeda 940 is an autologous program using the 1XX co-stim domain. And what's impressive about this program is that the overall response rate is close to 90%. Even more impressive than that is the complete response rate is over 70%. This compares very favorably with the approved autologous CD19 therapies on the market right now. What's notable is that 1XX allowed the Takeda scientists to identify a recommended phase 2 dose of 25 million T cells. And that's really impressive because typically the approved CAR T therapies on the market right now typically use 200 to 300 million CAR T cells. We feel like this contributes probably to safety, but also efficacy. And one other data point from the clinic is that we know now increasingly that less differentiated T cells are probably essential for durability and response in patients. This was validated by Novartis on their YTB323 program. This was a stem-enriched autologous CD19 CAR T program. It's basically identical to the Kymriah approved product, but it uses a shorter manufacturing process to enrich for stem-enriched T cells. And here, the difference between Kymriah is also notable. Here with YTB323, the complete response rates also appear to be greater than 70%. And these complete responses appear to be durable at 6 months. Also notable is that their recommended phase 2 dose is 12.5 million T cells. Again, this is 1/20th the amount of CAR Ts in the approved drugs you see in the market. So based on these clinical data, we're very excited about ATA3219. Just to pause and remind you where we are as we pivot to CAR Ts. Our first program is 3219. This is a CD19 targeted chimeric antigen receptor. And our other program we'll touch upon is 3431, which targets CD19 and CD20. And we'll walk you through some preclinical data for why we feel like these programs are differentiated and probably could be best in class. When we think about the 3219 opportunity in non-Hodgkin lymphoma, we still feel that there's an opportunity to compete because it's a differentiated CAR T product. There's already been proven limitations in this space with other CD19 targeted therapies. The first challenge in the space is really access. So what time has shown is that while this could be already a blockbuster drug, you're still not able to reach the majority of the patients. You might be reaching maybe 20 to 40% of the patients. And that's really because largely autologous CAR T cell therapy has really been limited to academic centers. So in other words, if you don't live within 50 miles of an academic center, you probably can't receive this amazing treatment. The next piece is durability, where we know that autologous CAR T therapy really at 6 months to complete response rates, really 30 to 40%. So if you think about it just from those 2 points that we teed up early, number one is efficacy. Number two is really access for patients and how you manufacture the drugs. When you look at what other modalities are out there, we still feel like bispecifics can be problematic in this space. Bispecifics may have a certain part of this market, but in terms of efficacy, bispecifics don't compare to T-cell therapeutics. And then when we zoom in on other allogeneic CD19 CAR T cell therapies, there's really been limited durability of responses. And that's why we still feel like there's an opportunity for us at Atara. Let me walk you through this data to highlight why we're excited about our program and why we think there's a potential for best in class still. So let me tell you about the model. This is a preclinical mouse model for CD19 positive malignancy. This is using a tumor cell line called NOM6, which is a leukemia. It's a very aggressive cell line. So if you start on the right of this slide, what we're doing is showing you efficacy compared to PBS control and non-transduced T cells. In red is really what we call an autologous CD19 CAR T benchmark. So in the labs, we recreated probably what is the best CAR T that's on the market right now. We compared to 3219, our EBV CAR T therapeutic. And really what I hope you see is the percent survival is really different. So like I said, this is a very aggressive model. By 20 to 25 days, all of the mice succumb to this aggressive tumor. And what you see even with the best auto CAR T is over time, your survival goes from probably 80% down to 20%. What we noted and what got us excited is that with 3219, we were seeing durable responses throughout the course of the study, well beyond 25 days. In addition, when we now focus on the middle of this slide, what we realize is that efficacy probably correlates with longer persistence of the 3219 CAR T cells. Again, these are allogeneic CAR T cells being compared to an autologous CAR T cell in red. And you can see by VCN, which is really detecting the CAR gene product in T cells in the blood of these mice, that you can see that our allogeneic CAR T cells persist in this model. And then what we're showing on the far left is really there's a notable difference in the phenotype. So most CAR Ts that are autologous look like what we're showing you here. It's not a memory phenotype. It's very much an effector T cell phenotype. And the reason why this is problematic, we feel like, is that these T cells exhaust pretty quickly. And you can see at 3219 on the far left is very much enriched, more than 90% for these central memory T cells. So in total, we think that central memory phenotype plus 1XX for persistence really result in this robust performance in this preclinical model. In addition to 3219, we have another CAR T program that we feel like we could file an IND for in the second half of next year or thereabouts. And this is really targeting two antigens. It's targeting CD19 and CD20. And we feel like this also not only has the ability to be best in class, but also a game changer. Targeting both antigens reduces the probability of antigen escape, which is something that we observe in the clinic for T-cell engagers as well as CAR T therapeutics. We also know that autologous CAR T programs have shown very promising efficacy. In fact, CAR 39, which is a program in-licensed by Janssen for $250 million, really is an impressive program. What you can see from their data are that probably by targeting both antigens on B-cell lymphomas, you have a response rate that's greater than 90% and a complete response rate. It looks like it's greater than 85%, at least in their data that they've shown. So imagine then taking this efficacy, but with an allo approach. So you reduce your cost of goods and you get this therapeutic to the patients faster. So we feel that also, again, with 3431, we have this amazing opportunity to really impact patients positively. So I'm going to walk you through the data for why we feel very strongly about this program also. So what this experiment is this is using the Raji tumor cell line. And what I'll tell you about the Raji model is that when you evaluate CD19 CAR T cells in this model, they don't work very well in terms of slowing down tumor progression. This is a very challenging model. It's one of the more challenging models preclinically that we have. It's CD20 positive, but it's also CD19 low. And what you can see here on the left is really you're looking at tumor burden by fluorescence. And what you can see is that an auto benchmark, in other words, a program that is in the clinic, we took that design, we recreated in the labs. You can see that the auto benchmark in red has a certain ability to control the tumor, but our off-the-shelf allo EBV CAR T cell program really shows you much better tumor control. And this is throughout the course of the study. And then when you pivot to the right side of this graph, this is percent survival. And again, you can see the survival advantage that 3431 has. So it's for this reason that we're really excited about this program also. So just to conclude, our CAR T programs support transformative potential. And we also believe going into autoimmunity is the next key area for us as a company. High unmet need. We've already seen proof of concept in lupus, but those were with autologous programs. And if you think about how many patients there are, it's probably impossible to attack this problem with an autologous approach. At best, you could probably make about 10,000 lots per year, but that really doesn't even touch the potential and the unmet need of 100,000 patients maybe with lupus nephritis. There's no allo CAR T product yet with clinical data. And we feel that with our proven safety already from tab-cel and our allo CAR Ts, we're pretty excited to also go into this area of unmet need. So I'm just going to conclude here. This is our pipeline, and thank you for your attention.
Thank you, Dr. Nguyen. It sounds like there are several advantages to your CAR T programs, your EBV T cell backbone, the various attributes that you're employing to optimize your cells, and the fact that they're allogeneic versus autologous. Is that right? And what would you say are the most compelling features versus your competitors?
So to answer the first question, what we're excited about is we've learned so much from tab-cel. And so we're probably already operating in a very scalable way. So we are very confident in our ability to make these CAR T cells. In terms of comparing to our competitors, we feel very strongly that the data show that gene editing might not be compatible with T cell biology. You start to see some worrisome trends. To date, most of the allo CAR T approaches have used gene editing, and they really haven't worked that well without aggressive lymphodepletion. And one of the issues there, Robert, is that what we don't talk about is aggressive lymphodepletion really correlates very strongly in month 3 with enhanced infections for patients. You see neutropenias and a lot of infections, and clinicians tell us time and time again, this is not good for the patients, and they'd really rather avoid lymphodepletion at all if they can. And in fact, what we've seen with tab-cel is we don't have lymphodepletion. So one of the areas that we are going to pivot eventually with our CAR T programs is we will do a level set and have a proof of principle in patients. And with clinical amendments, we will then also go forward without lymphodepletion.
Yeah, it sounds like avoiding gene editing would be an advantage in cell therapy for the reasons you just mentioned.
Yeah, and I think we're talking about the patient experience, but also there's a manufacturing cost, Robert. Gene editing right now as it stands is very inefficient. So in its most efficient guise, when you have 2 edits, right, and it's really the probability of success of the 2 edits is 70% times 70%, you really have only about 50% of your manufactured T cells with the correct gene edits. Over time, that really hurts 2 things. Number one, your ability of the T cells to proliferate so that you can have enough T cells for your productive GMP lot. But the second is it really, you take a big hit on your cost of goods. It's really inefficient.
Well, the cost savings for your method would make it not only more cost effective to treat the patients, but one of the things that you touched on was that patients have to live near an academic center.
Yeah.
And what you're describing sounds like this is easier to administer by routine medical practitioners. So you would be not only reducing the cost, but you would be opening up the field and the therapy to more patients.
Yeah, we'd agree with that characterization. With our remarkable clinical experience in 600 patients, we really haven't seen a safety signal. So I think that will probably give clinicians confidence to really join our clinical trials, treat patients with these CAR T products.
Yes, and have you spoken with physicians about the difference between the autologous and the allogenic, and why the allogenic might be better from their perspective?
Yeah, that's another thing that we don't talk about. When you have an autologous CAR T therapeutic, they are showing remarkable efficacy. Right, that's Tecartus and Yescarta in lymphoma, and Carvykti is spooling up to be a $5 billion drug in myeloma. But patients have to go on bridging therapy. So what you're asking is a patient whose tumor has come back pretty aggressively to now go on chemotherapy for 30 days or so and wait for a CAR T. That's asking a lot for a patient. That's why we think allo, for many reasons, is the answer. So not only can you, and what we've shown on Tab-cel is this ability to get product to patients within 3 days. We learned this globally in Europe and the United States, how to get Tab-cel into patients, 15-minute infusion. We feel like using the same paradigm and the same experience we have with global logistics will help us to get these medicines to patients faster.
Well, it sounds almost as if you have a new technology that's superseding what's out there now in both efficacy, safety, administration, and the outcome for the patients. So,
yeah.
Okay, if I could just go back to some of the science, how do you accomplish the HLA matching, and what exactly does that process involve, and just reiterate some of the importance of it?
Yeah, so I think we've used HLA, partial HLA matching in Tab-cel for over 20 years. And what's interesting is Tab-cel was invented. The problem statement was patients post-transplant were dying of lymphomas. And the only answer at the time was donor leukocyte infusion, which was pretty crude. Graft-versus-host with that drug was 40 to 60%. So Memorial Sloan Kettering invented Tab-cel as an allo solution so that you could be in time to save these patients. And what they found was that you only needed 2 out of 10 HLA matches for efficacy. And really, that's been amazing over the 20 years to have this consistent 50% efficacy. And well, I'll tell you what is interesting to us is recent data from others have shown in one of the CAR T studies, I believe this was Caribou, they also feel like there was a patient benefit with their allo CAR T therapy. When they cut the data, they realized that there was better efficacy when there was minimal HLA matching. So we feel like not only in our experience with Tab-cel, but also this orthogonal data set from Caribou, it really tells us that we're moving in the right direction. And furthermore, if you can achieve that without all the gene editing, we feel like really that makes a more robust T-cell product, Robert.
Yes, I would agree with you on that. Okay, and could you just take a moment to recap some of the key catalysts and milestones coming up for the company?
Eric?
Yeah, thanks for the question, Robert. Yeah, I think just in order of timeline, we have the BLA filing for Tab-cel coming up in Q2. So really, any minute now, we're hoping. And then we've said for 3219, in terms of NHL data, that's going to be Q4 this year. So we'll get a preliminary data there to see a signal for NHL. And then for lupus data, it would be first half of next year for prelim data. So that's how we're kind of moving along. I think, also early next year would be a hopeful BLA approval for Tab-cel. And with that, our partner, Pierre Fabre, our global partner, Pierre Fabre, would take over all of Tab-cel activities from regulatory development, manufacturing. They're already going to commercialize the drug. And then we're entitled to significant double-digit royalties when that happens. So it's a big transition for us as a company. And as I mentioned, kind of early 2025 time period at BLA approval.
Great. Okay, well, Dr. Nguyen, thank you very much for the presentation. Eric, great to see you again, and wish you the best in all of these milestones. Thank you.
Thanks, Robert.
Thank you.